Another screencast. I'm going to deal with the electron transport chain and oxidative phosphorylation. So in order to really understand this, you need to have a grasp sort of on facilitated diffusion, this energy coupling concept, redox reactions, and the structure of the mitochondrion. By the end of this, you should be able to describe the reactants, products, location, and purpose of the electron transport chain and oxidative phosphorylation. And to do that, to show an understanding of this, you should be able to explain how NADH is used in creating a proton gradient. You're going to be able to explain the role of oxygen, explain what chemiosmosis is, as well as then oxidative phosphorylation. So let's get into this. We've been using this as our guideline through here, and we're going to be addressing this part of the diagram here. So this is the third part. Other screencasts have dealt with the rest of this diagram. So this is where we are on our map. Electron transport chain, oxidative phosphorylation. So in the upper right here, let's just take a look at the mitochondrion again. Quick review. Cytoplasm matrix is the yellow. The intermembrane space is the orange. And that intermembrane space is separated from the matrix by the inner membrane of the mitochondrion. And this is what's important here. So if we go down to the larger diagram, the inner mitochondrial membrane is here. And remember, membranes are separating fluids, keeping things that are large, charged, and polar from going across that membrane. So let's orient ourselves. This is the intermembrane space. And then here's the mitochondrial matrix where the Krebs cycle is occurring. So since the Krebs cycle is occurring in the matrix of the mitochondrion, we're going to have this NADH being produced here. Let's just look at this membrane again here real quick before we get started. So here's the inner membrane of the mitochondrion, and wedged in that membrane there are these protein complexes, right? And this protein complex, where there's this one, there's also this one and this one and this one. So these are not found separate from one another. These protein complexes of electron carriers are called cytochromes. Cytochromes. It's not important that you know the name of each individual one, but just that they are cytochromes. And they are these proteins wedged in the membrane, and they are going to be responsible for transporting the electrons. So this is an electron transport chain. 1, 2, 3, 4, 5 cytochromes in the membrane. I'm going to drop a line down here and also point out that there's another protein in these membranes, and this protein is Atp synthase, and this Atp synthase is an enzyme. So this ATP synthase is separate from the electron transport chain, right, but also found in the inner membrane of the mitochondrion. Let's go back to see what's happening here. So in oxidative phosphorylation, we're going to oxidize something. What's going to be oxidized is NADH. So in oxidative phosphorylation, we're going to oxidize NADH, and oxidations are negative delta G. So when I oxidize NADH back to NAD+, this is going to be a negative delta G reaction. So I'm going to oxidize it. What's going to oxidize it? Well, these cytochromes are going to be reduced. So these cytochromes oxidize NADH. So as the electron in yellow here, right, it is donated to this cytochrome. This cytochrome is said to be reduced, and this NADH is said to be oxidized. Then this cytochrome is going to transfer or transport this electron to the next cytochrome in the chain. So then we oxidize this cytochrome and reduce this cytochrome. So as this redox happens here, as this redox happens and as this redox happens, these are all negative delta G, so we're oxidizing the whole time. So as we oxidize, that's negative delta G. We're going to couple this positive delta G reaction right here of moving 2 hydrogen ions or 2 protons, right, into the intermembrane space. So this is going against the gradient. That's why this is a positive delta G reaction. We are making a proton gradient here. These oxidations give us the energy to make that proton gradient. Just a quick aside, again, a proton is a hydrogen ion. These are the same things. Remember that a hydrogen is 1 proton and 1 electron. So if I take the electron off of that, I'm left with just a proton, right? And that is a hydrogen ion. So what we're making here is a proton gradient. Proton gradient, or hydrogen ion. gradient. So this cytochrome right here can also be thought of as a proton pump. It's moving protons against its gradient, and concentrating it in this intermembrane space. So as these electrons are transferred from one cytochrome to the next, and one cytochrome to the next, and one cytochrome to the next, all these redoxes are negative delta G. So the movement of these electrons through the chain is a negative delta G. That provides the energy to drive this positive delta G reaction. Here it happens again. And then it happens again at this very last step with this cytochrome in the chain. So each time electrons are moved, protons are pumped. Kids will screw this up. They'll write protons and electrons and think they're the same thing, and they're different from one another, okay? So protons get pumped, electrons get moved. So as the electrons are transported, the protons get pumped. Electrons move, protons get pumped to make this proton gradient. At the end of the chain, what is accepting these electrons at the end of the chain? What's accepting it is oxygen. So these oxygen atoms here are, you can also think of that they are pulling the electrons off of that cytochrome there. Right? Oxygen is extremely electronegative. This oxygen is going to be oxidizing this last cytochrome in the chain. And when it does that, it's reduced to water. Now, you could just have this oxygen, oxidize this NADH directly, right? And then you could do this way. But then what don't you do? If you just do that direct redox right there, you don't move electrons through the chain. And if you don't move electrons through the chain, what don't you do? You don't pump protons. So pumping these protons, making this proton gradient is pretty dang important to this whole process right here. So by having oxygen at the end, right, this is the oxidation part of oxidative phosphorylation. Now, where's the phosphorylation part? Well, the phosphorylation part is over here on the right, where ADP and inorganic phosphate are going to come together and form ATP. So this is the phosphorylation bit over here. Right? Now, how does that happen? Well, this enzyme, ATP synthase, makes that happen, but it doesn't just make it happen on its own. What's going to happen is this proton gradient over here is going to be utilized, right? And two protons at a time diffuse down through, right, or back across the membrane through ATP synthase. So this is a channel as well as an enzyme, and that enzyme is activated in some way to phosphorylate this ADP. So the movement of these protons back across the inner membrane of the mitochondrion is referred to as chemiosmosis. The facilitated diffusion of protons is chemiosmosis. Osmosis, I know you're thinking like, oh, water's moving, but it's not water moving, it's going to be a chemical moving. What's the chemical? The chemical is these protons here. So in order to have osmosis, in order to have facilitated diffusion, I have to have a gradient. Well, how did I get the gradient? I got the gradient because I moved electrons from NADH to oxygen. And as I moved those electrons, I pumped protons into the intermembrane space. Now over here, I'm going to utilize that gradient, and I'm going to allow them to facilitatively diffuse back in. And as I do that, I can generate ATP. So for every one molecule of NADH that goes in here, how many protons do you pump? 2, 4, 6. Well, if I pump 6 protons, how many ATPs can I make? Well, if the 2 that come back in make 1 ATP, and I've got 6 up here, I could do 2 is 1, and another 2 is 4, no, it's 2, and a third set of 2 is 3. So for every one molecule of NADH, 1 NADH, this equals 3 ATP. So that actually is a pretty decent payoff, right? So there is electron transport chain, oxidative phosphorylation. Again, the oxidative part is all oxidizing NADH. The phosphorylation part is over here. So as you oxidize, you generate proton gradients, and then you phosphorylate via chemiosmosis. Here's a little, I would go watch this little animation here to show you the activity of ATP synthase. What's essentially happening, right, again, is that you have this proton gradient. Protons are in high concentration here. Right? Protons are in low concentration here. And as the protons diffuse back across chemiosmotically, as the protons diffuse across, that provides the energy for this thing to spin around and do some biochemistry magic here, and you make this ATP. So there's a little bit more detail if you're interested. Lastly, we'll put these on this energy gradient. So here's the free energy gradient here. And we're looking at NADH at a relatively high free energy. And here's water down here at a really low free energy. But this is all relative to oxygen. So these are supposed to be the cytochromes in the chain. Right? So these are all cytochromes. And the structure of those cytochromes makes them themselves have a free energy gradient there. But when I reduce it, and then when I oxidize it, as these electrons move, they're moving down an energy gradient. So as the electrons move this way, they're moving down an electron gradient. So sometimes you'll hear this or read this as, that the electrons are falling down a gradient, that they are cascading down an energy gradient through the electron transport chain. Right? So this is electron transport chain. And as they fall or cascade down, of course, this is referring to the fact that it's a negative delta G, right? And this negative delta G here is going to be coupled with the positive delta G of doing what? Right? When these electrons move, what's the positive delta G that we do? Make ATP? Yeah, sort of, kind of, but the better answer is that this positive delta G coupled to this negative delta G is making a proton gradient. Right? Then that proton gradient will then later be used in chemiosmosis to generate ATP from ADP and inorganic phosphate. That's really sloppy. But there's a blank diagram to kind of sort of fill in. This, again, is my creation. Yours may look different, but there we go. All right. Hope that was helpful, and we'll see you in class. Oh, if you want to just take a look at this, right? How is this an analogy to the electron transport chain? So when I say how is an analogy, you should be able to say, like, okay, what is this? What does this represent? What does the water represent? What's this? Right? What are all these parts? If this was electron transport chain, what would those different parts be? You know, what would this be over here? Right? So you can practice that. Hope that was helpful. Bring questions to class.